I'd like to now tell you about how data are interpreted across the country by most crime labs that haven't brought this sort of computer technology on board, and what's been going on for the last 15 years. The original case reports for mixtures that crime labs produce don't have typically numbers that are at the same level for single source DNA as they do for mixtures, because they tend to not separate them out. If you're not separating out a mixture, what are you left with? So let me show you that. Instead, what most DNA analysts do in the US is they look at the peaks and they make a summary of the data that they're seeing. They don't look at the highs and the lows. They don't look at the patterns of what might go together, like the two tall peaks, the two small peaks. They don't assess the variation between the peaks. Computers can do that. They can model all of that, the patterns, the variations, the quantities. What people do in a much simpler procedure that the FBI rolled out maybe 15 years ago to make it easier for analysts to get to analyze their data was that the laboratory determines a threshold, and the threshold here is just a peak height, say 50 or 100 or 200. I think in this case a level of 50 RFU were used. And then all the data in their lab, it's like you're drawing a line through the peaks and you're asking, am I over the threshold or under the threshold? Okay. We can talk about whether that's a good idea or not later on, but that's the procedure that's being done. And in this case, if you would determine that these 4 alleles are all over the threshold, then if there are 4 alleles, the 8, 11, 12, and 14, there would be 10 pairings of those 4 alleles. And so instead of, with a computer, being able to focus the probability all at the 12, 14 answer, the probability gets dispersed across, against 10 possibilities. And that loses information. On the other hand, has a very simple formula. This is the only math I'm going to show you, but I'm just adding things together and multiplying. The probability of inclusion, or PI, we're going to calculate now at the D13 locus. It's a very simple formula. The FBI provides PopStats software for this. Again, these alleles, anything over the threshold is called an allele. It doesn't mean it actually is an allele. It might be an artifact of some kind. But for convenience, suppose it's an allele. That's why it's in quote. For all these putative alleles over threshold, add up their frequencies. And here are the frequencies of 8, 11, 12, and 14. That is how common they are in the population, or how rare. Get the sum, square it, and get a number. That's the probability of inclusion. And then the strength of match for this threshold method is 1 over that rarity. And that number in this case is 1.71. Okay, so it's bigger than 1. That's showing some support for an inclusion. But the computer's match statistic at this location was 35. And if you're going to multiply numbers together, if you're multiplying numbers like 1 and 2 together, as opposed to numbers like 10 or 20 together, you're going to get smaller weights of evidence when you're done. And that's what we see. So this is the same picture I showed before, but now I've added in red what the match statistic is for 12 of the 13 loci that they analyzed. At the locus D7, the suspect was under the threshold, so they didn't use it, so we just put a 1 in there. But you can see that the combined probability of inclusion, combining these 12 probabilities of inclusion and multiplying them together, each number shown in red using this method is a lot, each number is smaller, and therefore the product is smaller, and this is the comparison of information when we're done. The method, here's the combined probability of inclusion that was in the lab's report, and here's the true allele result that was in cybergenetics report. On the genes that we saw, whereas we've reported 2 quadrillion, which has 15 zeros after the 1. Quadrillion's a 1 followed by 15 zeros. The CPI method, with these thresholds, produced a number in the hundreds of thousands, which has 5 zeros. 1 followed by 5 zeros is 100,000. And we saw the same sort of result on the T-shirt. It's partly because in the genes it was this is a 10% mixture, and on the T-shirt it was a 25% mixture, so they're low amounts of a minor contributor. So on the T-shirt, the original match statistic is 5 zeros after the 1, 630,000. And again, quadrillion is 15 zeros. The reason I'm showing you these zeros is this is how scientists, including Alan Turing, measured information. In fact, he came up with a unit for measuring information. You take the number of zeros in the match statistic, the likelihood ratio, that's the amount of information. So if it's a quadrillion, the information's 15. If it's 1,000, 1 followed by 1,000, 3 zeros, the information's 3. And so for the rest, much of the rest of what we'll be looking at, I'll be showing you what's called the log of the likelihood ratio, the base 10 logarithm. I'm not going to say that. It's the number of zeros after the 1 in the number. Okay? What do you do if the number's between 100 and 1,000? Well, there's 2 zeros in 100, there's 3 zeros in 1,000, so the log is some 2 point something. So that's where these information numbers come from. They're the magnitude, the powers of 10 of match statistics. Okay, so I'm going to make a quick comment, which will come up repeatedly, about validation. TrueAllele is an extensively validated genotyping method. There have been dozens of studies that have been done by ourselves and by laboratories that are using it or bringing it on board. And some of these studies, so far, 7 of them, have been or are about to be published in peer-reviewed scientific journals. What is peer review? The concept of peer review is that our team of scientists, usually more than one person, doesn't have to be, they do a study, they prepare it, they work out what their methods are, their materials, they apply the methods to the materials, they obtain results, they write a scientific paper, they send it to a journal. And then the editor of the journal sends that out to 2 anonymous reviewers who are peers in the field, and they, usually your competitors, like if you're in academics, it's whoever you're competing with grant money against. So people who would have an interest in reviewing it, and then they look at it and they make comments about what they think of the study, how it could be improved, whether to reject it, accept it, make it better, and so on. Those comments go back to the authors, who then typically will make the improvements if the paper hasn't been rejected. This can go on for a few rounds, and ultimately, through this independent, anonymized peer review process, papers get accepted and published in scientific journals. You don't just send it in and it appears. Some papers might get accepted in a month or 2, others may take a year or 2. It depends on the journal, the review, the study, and so on. So the first studies that we were publishing were back in 2009. PLOS ONE is an open access journal, and that talked about the gap between human and computer interpretation. There was a paper, so I want to make a distinction. The top 4 articles that are shown in a lighter blue, these are done on samples of known composition that are synthesized in a laboratory. The bottom 3 were all done on casework samples. And the reason it's done both ways is that way you know what's happening under both conditions. Theoretically, you know more about the data if you synthesized it in the lab, but real casework has data that's a lot uglier than what you synthesize in the lab. And so you want to make sure your systems can handle both. So without going through them all, the top 4 were done on samples of known composition, and I'll make some comments about the bottom 3 that were done on casework. Most of these are available on our website or from the journals. We try to pay open access fees so anybody can download them. If you're interested in getting a copy of any of these articles, and because of copyright restrictions, I can't post them on the website, email me. My address will be at the end. I'm happy to send you whatever you want to read about. Let's take a look at the first New York State study that was done with the New York State Police as they were getting their TrueAllele system ready. This is done in collaboration with Dr. Barry Duesman, who recently retired. So it's a joint effort between cybernetics and the New York State Police Lab. One of the first observations is that the computer preserves more information. In short, what happened is we looked at 8 cases where of 2-person mixtures, like the ones we saw, where they reported an answer, so they got an answer. And then their question is, given that the lab got an answer, what would the computer do? That was the part of the study. Shown in red or orange are the number of zeros. So now we have the number of zeros on the y-axis here. Right, 5 zeros is 100,000, 10 zeros is 10 billion, and so on. We're measuring information in the number of zeros. And what we see is the average statistic for human review was about 7 zeros or 10 million. The average for the computer review was about 6 zeros more, about 13 million. And in this particular study, it doesn't have to be this way, the computer was able to extract more information from the same data that the lab had reported on. In a paper we did with New York State 2 years later, we looked at a lot of other questions on a much larger sample set. This is over 80 items of evidence. And again, in orange, we're now looking at the same inclusion method statistic, but again, the same statistic of around 6 or 7, a million or 10 million. The inclusion method pretty much always gives you the same answer. It's always around a million or 10 million if you're looking at around 13 or 15 loci. And in blue, this is the match statistic that the computer found. So now we're turning the question around. We're saying the computer looked at all of these items, and here's the information. And the scale here goes from 0, which is no information, up to over 20, which is approaching a trillion. And here, this line is showing how much information the computer got on these 85 comparisons. The average is about 15 zeros, and it has a number on all of them. And the most telling thing about this picture is regardless of the method used, the inclusion method in orange, or assuming the victim was known in green, or assuming it's not even a mixture in gray, is that these bars represent only 30% of the items. So one of the things that became clear early on is that whereas the computer is extracting information from all of these evidence items, people were only reporting in this study at that lab on only 30% of the evidence. So all the work was done, the data was generated, but stats weren't being reported and the data wasn't being used in criminal justice. In a study I'll come back to, which is done with Virginia, if you're going to read or look over one paper, this is the one I'd recommend. It was published last year, 2014. Here's the title of it. It's open access and you can read it. This is a major study that was done on Virginia casework data. And here is the range of information that they obtained. Again, you see that on a scale of 0 to 25, in other words, a number of 1 to a trillion. We're looking at the number of zeros. The amount of information is pretty much concentrated around 6 or 7. It's in the millions. You always get that answer with, typically, if you use all the locations with CPI. And so this is combined probability of inclusion, the standard human review method. And so this is the information they got, but problems began percolating in the forensics community around 10 years ago. Dr. John Butler at NIST, the National Institute of Standards and Technology, started doing interlaboratory studies. For example, he took a simple 2-person mixture, sent it out to 60 laboratories, and had them all interpret it and send back what their match statistics were. And this is his slide. As it says, they're all looking at exactly the same signal, those peaks that are going up and down, the electropherograms. And many groups said they couldn't interpret it. Of the groups that did, the amount of information ranged from 4 zeros in the tens of thousands up to 14 zeros, or hundreds of trillions. Same data, 10 billion-fold variation. 10 billion is 10 zeros in it. 10 billion-fold variation of what the interpretation is using mainly these threshold methods and inclusion methods. So that created some concern. As a result, the FBI Scientific Working Group on DNA Analysis Methods, or SWGDAM, came up with some guidelines hoping to reduce this variation across all the laboratories. And their suggestion was have a second threshold, right? We had 1, let's have another one. And so now in addition to the lower one, they raised it up and said, well, maybe if we have a threshold, it turned out to be about 3 times higher in the different labs, called a stochastic threshold, that'll account for more of the variation. Well, one of the problems with it is now you're throwing out the baby with the bathwater. In this case, that's Mr. Skundrich, and his DNA has just disappeared. And so you're not using it. It's below the stochastic threshold. But that wasn't the only problem with stochastic thresholds. You lost a lot of information. The average match statistic, you went from being able to interpret most of your evidence to not being able to interpret most of your evidence. The numbers went down. And at the same time, the average information went from the millions to the hundreds. That number, 140, is the average match statistic in our study, looking at when Virginia looked at 50 evidence items that were very low, that were challenging samples. And the reason that happened is that in the study, they ended up throwing out 2 out of every 3 locations. And so the match statistics dropped from the millions to the hundreds. And then there was another problem. NIST came back in with another study. This is their MIX13 study, meaning mixtures in 2013. This time, 100 groups wrote back on 5 different mixtures. And this is a 3-person mixture, which does not contain suspect C. He's not a contributor to this mixture. And of the 100 replies they got back, 70 labs said he was in the mixture. So now we're starting to get false inclusions that the federal government is warning the crime labs about, saying you've got to start doing something. 6 groups did give an exclusion. One of them used TrueAllele. But the range of what these statistics were are still varying from 9 to 350,000. So they're falsely including in a 3-person mixture, and at the same time, the numbers are still all over the map.